Hi, everyone. Welcome to Module 8.2.1. Remember that the rest of this module is just going to be further information about what the overview was for Module 8.1. So in this one and the following ones, you're going to see some information that we've already talked about. So you may not need to listen to this whole thing because 8.2.1, 8.2.2, and 8.3 are going to be significantly more detailed than 8.1 was, which is going to mean a longer video and a longer PowerPoint. It would still benefit you to go through all of this and know this material, as well as modules 9.2.1, 9.2.2, 9.3, and 10.2.1 and 10.2.2. So let's go ahead and jump into it here. So as we've already talked about, the adult human brain contains about 97% of the body's neural tissue. So a typical brain weighs about 3 pounds, has volume of about 1,200 milliliters, and brain size varies considerably along individuals. Like I said in the previous module, brains of males are on average about 10% larger than females, but that's due to differences in body size. And again, no correlation exists between brain size and intelligence. And we know that because the smallest brains, which are about 750 milliliters, and the largest brains, which are about 2,100 milliliters, are functionally normal. So there are 6 major regions of the brains. We have the cerebrum, the cerebellum, the diencephalon, the midbrain, the pons, and the medulla oblongata. The largest portion of the adult brain is the cerebrum. It's made up of large paired left and right cerebral hemispheres. So the brain has an extensive area of cortex, a superficial layer of gray matter covering most of its surface. So the surface of the cerebral hemispheres are highly folded and covered in a neural cortex called the cerebral cortex. And the cerebral cortex forms a series of elevated ridges or gyri, and gyri increase its surface area. Gyri are separated by shallow depressions called sulci or by deeper grooves called fissures. The cerebrum is the seat of most higher mental functions. Conscious thoughts, sensations, intellect, memory, and complex movements all originate in the cerebrum. And the cerebellum is the second largest part of the brain. It is partially hidden by the cerebral hemispheres. And like the cerebrum, the cerebellum has hemispheres covered by a neural cortex called the cerebellar cortex. The cerebellum adjusts ongoing movements by comparing arriving sensations with previously experienced sensations, allowing you to perform the same movements over and over again. Other major anatomical regions of the brain can be best examined after the cerebral and cerebellar hemispheres have been removed. And the walls of the diencephalon are composed of the left thalamus and the right thalamus. And each thalamus contains relay and processing centers for sensory information. The hypothalamus is the floor of the diencephalon and contains centers involved with emotions, autonomic function, and hormone production. The infundibulum, which is a narrow stalk, connects the hypothalamus to the pituitary gland and part of the endocrine system. The hypothalamus and the pituitary gland integrate the nervous and the endocrine systems, and the diencephalon is a structural and functional link between the cerebellar hemispheres, or the cerebral hemispheres, and the brainstem. The brainstem contains a variety of important processing centers and nuclei that relay information headed to or from the cerebrum or the cerebellum. And the brainstem includes the midbrain, the pons, and the medulla oblongata. So the midbrain, or the mesencephalon, contains nuclei that process visual and auditory information and controls reflex triggered by these stimuli. For example, your intermediate reflexes response to a loud, unexpected noise, such as eye movements and head turning, are directed by nuclei in the midbrain. This region also contains centers that help maintain consciousness. The pons of the brain connects the cerebellum to the brainstem. Pons is Latin for bridge. And in addition to tracts and relay centers, the pons contains nuclei involved with somatic and visceral motor control. The medulla oblongata connects the brain to the spinal cord, and near the pons, the posterior wall of the medulla oblongata is thin and membranous, and the inferior portion of the medulla oblongata resembles the spinal cord, and that has a narrow central canal. The medulla oblongata relays sensory information to the thalamus and to the centers and other portions of the brainstem. The medulla oblongata also contains major centers that regulate autonomic functions such as heart rate, blood pressure, and digestion. The embryonic origins of the brain can help us understand its internal organization. So the CNS begins as a hollow cylinder known as a neurotube, and this tube has a fluid-filled internal cavity called the neurococle. And in the cephalic portion of the neural tube, three areas enlarge rapidly through expansion of the neurococle. This enlargement creates three prominent divisions called the primary brain vesicles, and they are named for their relative positions. So the prosencephalon, or the forebrain, the mesencephalon, or the midbrain, and the rhombencephalon, or the hindbrain. So what you see here, table 14.1 summarizes the fates of these primary divisions of the brain. The prosencephalon and the rhombencephalon are subdivided further, forming the secondary brain vesicles, and the prosencephalon forms the telencephalon and the diencephalon. The telencephalon ultimately forms the cerebrum of the brain, and the walls of the mesencephalon thicken, and the neurococle becomes a relatively narrow passageway, much like the central canal of the spinal cord. The portion of the rhombencephalon adjacent to the mesencephalon forms the metencephalon. The dorsal portions of the metencephalon later become the cerebellum, and the ventral portions develop into the pons. The portion of the rhombencephalon closer to the spinal cord forms the mesencephalon, which becomes the medulla oblongata. So during development, the neurococle within the cerebral hemispheres, the diencephalon, the metencephalon, and the medulla oblongata expands to form chambers called ventricles, and cells of the ependymal line the ventricles. Each of the cerebral hemispheres contains a large lateral ventricle. The septum paludosum, which is a thin medial portion, separates the two lateral ventricles. Because there are two lateral ventricles, the ventricle and the diencephalon is called the third ventricle. The two lateral ventricles are not directly connected, but each communicates with the third ventricle of the diencephalon through an interverticular foramen. The midbrain has a slender canal known as the cerebellar aqueduct, and this passageway connects the third ventricle with the fourth ventricle. The superior portion of the fourth ventricle lies between the posterior surface of the pons and the anterior surface of the cerebellum. The fourth ventricle extends into the superior portion of the medulla oblongata, and there this ventricle narrows and becomes continuous with the central canal of the spinal cord. The ventricles are filled with cerebral spinal fluid, and the CSF continuously circulates from the ventricles and the central canal into the subarachnoid space of the surrounding cranial meninges. And the CSF passes between the interior and exterior of the CNS through three foramina and the roof of the fourth ventricle. The delicate tissues of the brain are protected from physical forces by the bones of the cranial meninges, and cerebral spinal fluid. In addition, the neural tissue of the brain is biochemically isolated from the general circulation by the blood-brain barrier. So the layers that make up the cranial meninges, the cranial dura mater, the arachnoid mater, and the pia mater, are continuous with the spinal meninges. However, the cranial meninges have distinctive anatomical and functional characteristics. The dura mater consists of an outer layer and inner fibrous layers. The outer layer is fused to the periosteum of the cranial bones. As a result, there is no epidural space superficial to the dura mater, as it occurs along the spinal cord. The outer periosteal and inner meningeal layers of the cranial dura mater are typically separated by slender gaps that contain tissue fluids and blood vessels, including large venous sinuses. The veins of the brain open into these sinuses, which deliver venous blood to the internal jugular veins of the neck. The arachnoid mater consists of the arachnoid membrane, which is an epithelial layer, and the cells of the fibers of the arachnoid trabeculae that cross the subarachnoid space into the pia mater. The arachnoid membrane covers the brain, providing a smooth surface that doesn't follow the brain's underlying folds. And this membrane, in contact with the inner epithelial layer of the dura mater, the subarachnoid space extends between the arachnoid membrane and the pia mater. The pia mater sticks to the surface of the brain, which is anchored by process of astrocytes, and extends into every fold and fissure. It also accompanies branches of the cerebral blood vessels as they penetrate the surface of the brain to reach internal structures. So we'll talk a little bit more about dural folds. In several locations, the inner layers of the dura mater extends into the cranial cavity, forming a sheet that dips inward and then returns. These dural folds provide additional stabilization and support to the brain. Dural sinuses are large collecting veins located within the dural folds, and the three largest dural folds are called the falx cerebri, the tendinorm cerebelli, and the falx cerebelli. So the falx cerebri is a fold of dura mater that projects into the cerebral hemispheres into the longitudinal fissures, and its inferior portions attach anterior to the cystogale of the ethmoid bone and posterior to the internal occipital crest, which is a ridge along the internal surface of the occipital bone. The superior sagittal sinus and the inferior sagittal sinus, two large venous sinuses, lie between the dural folds, and the posterior margin of the falx cerebri intersects the tendinorm cerebelli. So the tendinorm cerebelli protects the cerebellum and separates the cerebral hemispheres from the cerebellum and extends across the cranium at right angles to the falx cerebri. The transverse sinus lies between the tendinorm cerebelli. The falx cerebelli divides the two cerebral hemispheres along the mid-sagittal line inferior to the tendinorm cerebelli. So the overall shape of the brain corresponds to that of the cranial cavity, and the cranial bones provide physical protection by cradling the brain, but also pose a threat to safety, and that it's countered by the cranial meninges and the CSF. The brain is like a person driving a car. So if the car hits a tree, the car projects the driver from contact with the tree, but serious injury will occur unless a seatbelt or an airbag protects the driver from contact with the car. So the tough fibrous dural folds act like a seatbelt to hold the brain in position, and the cerebrospinal fluid in the subarachnoid space acts like a bumper by cushioning the brain against sudden jolts and shocks. Cranial trauma is a head trauma resulting from impact with other objects, and each year in the United States, about 8 million cases of cranial trauma occur, but only 1 case in 8 results in serious brain damage or a TBI. The percentage is relatively low because the cranial meninges and the CSF are so effective in protecting the brain. So cerebrospinal fluid completely surrounds and bathes the exposed surfaces of the CNS. The CSF has several important functions, including the following, which is cushioning delicate neural structures, supporting the brain, and in essence, the brain is suspended inside the cranium and floats in the CSF. So a human brain weighs, as we already talked about 1,400 grams in air, but only about 50 grams when supported by CSF. The third function is also to transport nutrients, chemical messengers, and waste products. So except at the carotid plexus, where the CSF is produced, the ependymal lining is freely permeable. So the CSF, in contrast with chemical communication with the interstitial fluid that surrounds the neurons and neuroganglia of the CNS. Because the free exchange between the interstitial fluid of the brain and the CSF, changes in the CNF function can produce changes in the composition of the CSF, which can be highlighted in about 5 steps. So 1, the carotid plexus produces and maintains the CSF. So 2 extensive folds in the carotid plexus originate in the roof of the third ventricle and extend through the intravesicular forma. These folds cover the floors of the lateral ventricles. In the inferior brainstem, a region of the carotid plexus in the roof of the fourth ventricle projects between the cerebellum and the pons. So 2, the CSF circulates from the carotid plexus through the ventricles and fills the central canal of the spinal cord. As it circulates, materials diffuse through in between the CSF and the interstitial fluid of the CNS across the ependymal cells. 3, the CSF reaches the subarachnoid space through 2 lateral apertures and a single median aperture in the roof of the fourth ventricle. 4, the cerebrospinal fluid then flows through the subarachnoid space surrounding the brain, the spinal cord, and the cauda equina. And 5, finger-like extensions of the arachnoid membrane, called the arachnoid villi, penetrate the meningeal layer of the dura mater and extend into the superior sagittal sinus. In adults, those extensions form large arachnoid granulations, and CSF is absorbed into the venous circulation at the arachnoid granulations. The carotid plexus produces CSF at a rate of about 500 milliliters, or about 2.1 cups per day. And the total volume of CSF at any moment is approximately 150 milliliters. So the entire volume of CSF is replaced about every 8 hours. And despite this rapid turnover, the composition of CSF is closely regulated and the rate of removal normally keeps about pace with the rate of production. So in forming CSF, a carotid plexus is an area within each ventricle that as we already talked about, produces the CSF, and specialized ependymal cells bearing microvilli and interconnected by tight junctions surround the capillary of a carotid plexus. So the ependymal cells secrete CSF into the ventricles. They also remove waste products from the CSF and adjust its composition over time. The differences in composition between CSF and blood plasma are quite noticeable. For example, blood plasma contains high concentrations of soluble proteins, but the CSF does not. The concentrations of individual ions and the levels of amino acids, lipids, and waste products are also different. In the normal circulation or reabsorption of CSF is interrupted, a variety of clinical problems can appear. For example, a problem with the reabsorption of CSF in infancy causes hydrocephalus or water on the brain. And infants with this condition have enormously expanded skulls due to the presence of abnormally large volume of CSF in that area. In adults, a failure of reabsorption or a blockage of CSF circulation can distort and damage the brain. So neurons have a high demand for energy, but they have no energy reserves in the form of carbohydrates or lipids. Neurons also have no oxygen reserves in the form of myoglobin. So your brain, with billions of neurons, is an extremely active organ with a continuous demand for nutrients and oxygens, and these demands are met by extensive circulatory supply. Arterial blood reaches the brain through the internal carotid arteries and the vertebral arteries. Most of the venous blood from the brain leaves the cranium in the internal jugular veins, which drain the dural sinuses. A head injury that damages the cerebral blood vessels may cause bleeding into the dura mater, either near the dural epithelium or between the outer layer of the dura mater and the skull. And these are serious conditions because the blood entering these spaces compresses and distorts the soft tissues of the brain. Cerebrovascular diseases are cardiovascular diseases that interfere with the normal blood supply to the brain, and the particular distribution of the vessels involved determines the signs and symptoms. And the degree of oxygen or nutrient starvation determines their severity. A cerebrovascular incident, or a CVA, or better known as a stroke, occurs when blood supply to a portion of the brain is shut off, and the affected neurons begin to die in a matter of minutes. Here you can just see some of the pictures of that venous return we just talked about. So neural tissue in the CNS is isolated from the general circulation by the blood-brain barrier. This barrier is formed by capillary endothelial cells that are extensively interconnected by tight junctions. These junctions prevent materials from diffusing between the endothelial cells. So in general, only lipid-soluble compounds, including carbon dioxide, oxygen, ammonia, lipids such as steroids or prostaglandins, and small alcohols, can diffuse across the membranes of endothelial cells into the interstitial fluid of the brain and the spinal cord. Water and ions must pass through channels of the apical and basement plasma membranes, and larger water-soluble compounds can cross the capillary walls only by active or passive transport. So the restricted permeability of the endothelial lining of the brain capillaries is in some way dependent on chemicals created by astrocytes. And these glial cells play a key supporting role in the blood-brain barrier because they release chemicals that control the permeabilities of the endothelium to various substances. Astrocytes are in close contact with CNS capillaries, and the process of astrocytes cover the outer surface of the endothelial cells. And if the astrocytes are damaged or stop stimulating endothelial cells, the blood-brain barrier disappears. The carotid plexus is not part of neurotissue of the brain, so no astrocytes are in contact with the endothelial cells there. Substances do not have free access to the CNS. However, because specialized ependymal cells create blood CSF barriers, these cells are also interconnected by tight junctions and surround the capillaries of the carotid plexus. Transport across the blood-brain barrier and blood CSF barriers is selective and directional. Even the passage of small ions such as sodium, hydrogen, potassium, or chloride is controlled. As a result, the pH and the concentrations of sodium and potassium, calcium, magnesium ions in the blood and the CSF are different. Some organic compounds are readily transported, and others cross only in very small amounts. Neurons have a constant need for glucose that must be met regardless of the concentrations in the blood and the interstitial fluid. And even when circulating glucose levels are low, endothelial cells continue to transport glucose from the blood to the interstitial fluid of the brain. In contrast, only trace amounts of circulating norepinephrine, epinephrine, dopamine, and serotonin pass into the interstitial fluid or the CSF of the brain. This limitation is important because these compounds are neurotransmitters. Their entry from the bloodstream where the concentrations can be relatively high, could result in the uncontrolled stimulation of neurons through the brain. The blood-brain barrier remains intact throughout the CNS, except in small areas known as circumventricular organs, or CVOs, which have fenestrated capillaries. And we'll talk more about fenestrated capillaries when you get into AMP2. Because they're outside the blood-brain barrier, they provide a direct link between the CNS and the peripheral blood vessels, and they play a role in the neuroendocrine function. So in summary, these noteworthy exceptions include, one, portions of the hypothalamus where the capillary endothelium is extremely permeable, and this permeability exposes the hypothalamic nuclei to circulating hormones and permits hypothalamic hormones to diffuse into circulation. Two, capillaries in the posterior lobe of the pituitary gland, which is continuous at the floor of the hypothalamus. At this site, the hormones such as antidiuretic hormone, or ADH, and oxytocin, produced by the hypothalamic neurons, are released into the circulation. Capillaries in the pineal gland, apologies, in the third summary, capillaries in the pineal gland. This endocrine gland is located on the posterior superior surface of the diencephalon, and the capillary permeability allows pineal secretions into the general circulation. And four, capillaries at the carotid plexus. The capillary characteristics of the blood-brain barrier are lost there, but the transport activities of specialized ependymal cells in the carotid plexus maintains the blood-CSF barriers. So keep in mind that physicians must sometimes get specific compounds into the interstitial fluid of the brain to fight CNS infections or to treat other neurodisorders. And in order to do this, they must understand the limitations of the blood-brain barrier and the blood-CSF barriers. For example, when considering possible treatments, the antibiotic tetracycline isn't used to treat meningitis or other CNS infections because this drug is excluded from the brain. In contrast, sulfazoles or sulfadiazines enter the CNS very rapidly, and sometimes chemotherapeutic drugs or imaging dyes may be injected directly into the CNS by lumbar puncture. The medulla oblongata is the most inferior of the brain regions. So in a sectional view, the inferior portions of the medulla oblongata resembles the spinal cord with a small central canal. However, the gray matter and the white matter organization is more complex. Ascending the medulla oblongata, the central canal opens into the fourth ventricle, and the similarity to the spinal cord all but disappears at that location. The medulla oblongata is a very busy place. So all communication between the brain and the spinal cord involves tracts that ascend or descend through the medulla oblongata. In addition, the medulla oblongata is a center for the coordination of complex autonomic reflexes and the control of visceral functions. The medulla oblongata includes three groups of nuclei. One, the autonomic nuclei controlling the visceral activities. The reticular formation is loosely organized mass of gray matter that contains embedded nuclei. It extends from the medulla oblongata to the midbrain, and the portion of reticular formation in the medulla oblongata contains nuclei and specific reflex or processing centers that regulate vital autonomic functions. These reflex centers receive inputs from cranial nerves, the cerebral cortex, and the brainstem, and their output controls or adjusts the activities of one or more of the peripheral systems. And there are two main groups of reflex centers in the medulla oblongata. The cardiovascular centers adjust the heart rate, the strength of cardiac contractions, and the flow of blood through the peripheral tissues. So in terms of function, the cardiovascular symptoms are subdivided into cardiac and vasomotor centers, but their anatomical boundaries are difficult to determine. The respiratory rhythmicity centers set the basic pace for respiratory movements, and their activity is regulated by inputs from the apneustic and pneumotaxic respiratory centers of the pons. So two, the sensory and motor nuclei of the cranial nerves. The medulla oblongata contains sensory and motor nuclei associated with the 5 cranial nerves, so 8, 9, 10, 11, and 12. These cranial nerves provide motor commands to the muscles of the pharynx, the neck, the back, as well as the visceral organs of the thoracic and perineal cavities. Cranial nerve 8 carries sensory information from receptors in the internal ear to the vestibular and cochlear nuclei, which extend from the pons to the medulla oblongata. And 3, they relay stations along the sensory and motor pathways. So the nucleus gracilis and the nucleus exocrinatus pass somatic sensory information into the thalamus. Tracks leaving these brain stem nuclei cross to the opposite side of the brain before reaching their destinations. This crossing over is called a decussation, and the site of decussation pyramids. Sensory information that reaches the CNS from spinal nerves and from cranial nerves. This information is integrated and forwarded to other autonomic centers in the medulla oblongata and elsewhere. So the olivary nuclei relay information to the cerebral cortex, my apologies, the cerebellar cortex, about somatic motor commands as they are issued by motor centers at higher levels. The bulk of the olivary nuclei creates olives or prominent olive-shaped bulges along the ventrolateral surface of the medulla oblongata. So the pons links the cerebellum with the midbrain, the diencephalon, cerebellum, and the spinal cord. The pons contains four groups of components. You have one, the sensory and motor nuclei of the cranial nerves, and these cranial nerves, 5, 6, 7, and 8, innervate the jaw muscles, the anterior surface of the face, one of the extrinsic eye muscles, which is called the lateral rectus, and the sense organs of the internal ear, or the vestibular and cochlear nuclei. Two, the nuclei involved in the control of respiration. On each side of the pons, the reticular formation in this region contains two respiratory centers, the aponestic centers and pneumataxis centers, and these centers modify the activity of the respiratory rhythmicity centers in the medulla oblongata. Three, the nuclei and tracts that process relay information sent to or from the cerebellum. The pons links the cerebellum with the brainstem, the cerebrum, and the spinal cord. And four, ascending, descending, and transverse tracts. Longitudinal tracts interconnect the portions of the CNS, and the middle cerebellar punctacles are located to the transverse fibers, which cross the anterior surface of the pons. And these fibers are axons that link nuclei of the pons, also called the pontine nuclei, with the cerebellar hemisphere of the opposite side. This is just a table that kind of highlights the information we just talked about. So the cerebellum is an autonomic processing center and has two primary functions. One, adjusting the postural muscles of the body. The cerebellum coordinates rapid automatic adjustments that maintain the balance and equilibrium. It makes these alterations in muscle tone and position by modifying the activities of motor centers in the brain. And two, programming fine-tuning movements controlled at the conscious and subconscious levels. The cerebellum refines learned muscle patterns such as riding a bicycle or playing the piano. It performs this function indirectly by regulating activity along the motor pathways at the cerebral cortex, the basal nuclei, and the motor centers in the brainstem. The cerebellum compares the motor commands with the proprioceptive information, so the position sense, and simulates any adjustment needed to make the movement smooth. The cerebellum has a complex, highly convoluted surface composed of neural cortex, and the folia, or the folds of the cerebellum surface, are less prominent than the folds of the surface of the cerebral hemispheres. The primary fissure separates the anterior and posterior lobes, and along the midline, a narrow band of cortex known as the vermis separates the cerebral hemispheres, and the cylinder focendicular lobe lies between the roof of the fourth ventricle and the cerebral hemisphere of the vermis. Like the cerebellum, like the cerebrum, the cerebellum has a superficial layer of neural cortex. The cerebellar cortex contains relatively huge, highly branched Purkinje cells, and the extensive dendrites of each Purkinje cell receive input from about 200,000 synapses, more than any other type of brain cell. So the internal white matter of the cerebellum forms a branching array and sectional view that resembles a tree. Anatomists call this the arbor vitae, or the tree of life. It connects the cerebellar cortex and the nuclei with the cerebellar punctacles, which we'll discuss here shortly. So the cerebellum receives proprioceptive or position information from the spinal cord and monitors all proprioceptive, visual, tactile, balance, and auditory sensations received by the brain. Most axons carry sensory information but do not synapse at the cerebellar nuclei, but pass through the deeper layers of the cerebellum on their way to the Purkinje cells of the cerebellar cortex. Information about the motor commands issued at conscious and subconscious levels reach the Purkinje cells indirectly after being relayed by nuclei in the pons or by the cerebellar nuclei embedded within the arbor vitae. Tracks that link the cerebellum with the brainstem, the cerebrum, and spinal cord lead the cerebellar hemispheres as the superior, middle, and inferior cerebellar punctacles. The superior cerebellar punctacles link the cerebellum with the nuclei in the midbrain, the diencephalon, and the cerebrum. The middle cerebellar punctacles are connected to a broad band of fibers that cross the ventral surface of the pons at right angle to the axis of the brainstem. The middle cerebellar punctacles also connect the cerebellar hemispheres with sensory and motor nuclei in the pons. And the inferior cerebellar punctacles communicate between the cerebellum nuclei and the medulla oblongata and carry ascending and descending cerebellar tracts from the spinal cord. The cerebellum can be permanently damaged by trauma or stroke. Drugs such as alcohol can temporarily affect it, and the result is ataxia. So it's a disturbance in muscular coordination. And in severe ataxia, the individual cannot sit or stand without assistance. This just highlights what we just talked about. So the external anatomy of the membrane is shown here. The tectum, or the roof of the membrane, is the region posterior to the cerebral aqueduct. It contains two pairs of sensory nuclei known collectively as the corpora quadrineum. And these nuclei, the superior and inferior cochlea, process visual and auditory sensations. Each superior colculus receives visual inputs from the lateral geniculate nucleus of the thalamus on that side. Each inferior colculus receives auditory input from nuclei in the medulla oblongata and the pons, and some of this information may be forwarded to the medial geniculate of the nucleus of the thalamus on that same side. So the superior coculi control the reflex movements of the eyes, the head, the neck in response to visual stimuli such as bright light. And the inferior coculi control reflex movements of the head, the neck, the trunk in response to auditory stimuli such as a loud noise. The area anterior to the cerebral aqueduct is called the tangentum. And on each side, the tangentum contains a red nucleus and the substantia nigra. So the red nucleus contains numerous blood vessels, which give it a rich red color. And this nucleus receives information from the cerebrum and cerebellum and issues subconscious motor commands that affect upper limb position and background muscle tone. The substantia nigra is the largest midbrain nucleus, and it lies lateral to the red nucleus. The gray matter in this region contains darkly pigmented cells, giving that kind of black color. And the pigment is melanin, and here it is a byproduct of dopamine synthesis. The substantia nigra inhibits activity of the basal nuclei in the cerebellum. The basal nuclei are involved in the subconscious control of muscle tone and word movements. As we'll discuss in a later section, Parkinson's disease is characterized by the loss of neuronal activity and the substantia nigra. So the nerve fiber bundles on the ventrolateral surfaces of the membrane are the cerebellar punctacles, and they contain 1 descending fibers that go to the cerebellum by way of the pons, and 2 descending fibers that carry voluntary motor commands issued by the cerebellar hemispheres. The midbrain also contains the reticular activating system, or RAS, which is a specialized component of the reticular formation. Stimulation of the RAS makes you more alert and attentive, and damage to the RAS produces unconsciousness. Again, just a table discussing what we just talked about. So the diencephalon is a division of the brain that consists of the epithalamus, the thalamus, and the hypothalamus. The epithalamus is the roof of the diencephalon, superior to the third ventricle, and the anterior portion of the epithalamus contains an extensive area of carotid plexus that extends through the interventricular foramina into the lateral ventricles. The posterior portion of the epithalamus contains the pineal gland. So that's an endocrine gland, or an endocrine gland that structure that secretes the hormone melatonin. Melatonin is important in regulating the day-night cycles and reproductive functions. Most of the neural tissue in the diencephalon is concentrated in the left thalamus and the right thalamus, which form the lateral walls, and the hypothalamus, which forms the floor. Ascending sensory information from the spinal cord and cranial nerves other than the olfactory tract, synapse in a nucleus at the right or left thalamus before reaching the cerebral cortex and our conscious awareness. The hypothalamus contains centers involved with emotions and visceral processes that affect the cerebrum, as well as other components of the brainstem. It also controls a variety of autonomic functions and forms the links between nervous and endocrine system. So, speaking specifically of the thalamus, on each side of the diencephalon, the thalamus is the final relay point for sensory information ascending to the primary sensory cortex. It acts as a filter, passing only a small portion of the arriving sensory information. The thalamus also coordinates the activities of the basal nuclei in the cerebrum and the cerebral cortex by relaying information between them. The third ventricle separates the left thalamus from the right thalamus. Each thalamus consists of a rounded mass of thalamic nuclei. Viewed in a mid-sagittal section through the brain, each thalamus extends from the anterior commissure to the inferior base of the pineal gland. A projection of gray matter, called the interthalamic adhesion, extends into the ventricle from the thalamus on either side, but no fibers cross that midline. So some of the functions of the thalamic nuclei. The thalamic nuclei deal primarily with the relay of sensory information to the basal nuclei, so the parts of the cerebrum where we'll discuss here shortly, and cerebral cortex. So the five major groups of the thalamic nuclei are the anterior, the medial, the ventral, the posterior, and the lateral groups. The anterior group includes the anterior nuclei, which are part of the limbic system, and this system is involved with emotion and motivation. The nuclei of the medial group provide awareness of emotional states by connecting emotional centers of the hypothalamus with the frontal lobes of the cerebral hemispheres. And the medial group also receives and relays sensory information from other portions of the thalamus. The nuclei of the ventral group relay information from the basal nuclei of the cerebrum and the cerebellum to somatic motor areas of the cerebral cortex. Ventral group nuclei also relay sensory information about touch, pressure, pain, temperature, and proprioception, or position, to the sensory areas of the cerebral cortex. The posterior group includes the pulvinar and denticulate nuclei, and the pulvinar nuclei integrate sensory information for projection to the cerebral cortex. And the lateral denticulate nucleus of the thalamus receives visual information other than the optic tract, which originates at the eyes. The output of the lateral geniculate nucleus goes to the occipital lobes of the cerebral hemispheres and to the midbrain. The medial geniculate relays auditory information to the appropriate areas of the cerebral cortex from specialized receptors of the internal ear. And lastly, the nuclei of the lateral group form feedback loops with the limbic system and the parietal lobes of the cerebral hemispheres. The lateral group affects emotional states and the integration of sensory information. This is just a table of what we just talked about. So speak a little bit more about the hypothalamus. So the hypothalamus extends from the area of the superior optic chiasm, so a crossover where the optic tracts from the eyes arrive at the brain, to the posterior margins of the mammillary bodies. So the mammillary bodies possess sensory information, including olfactory sensations, and contain motor nuclei that control reflex movements associated with eating, such as chewing, licking, and swallowing. They are also involved in memory. Immediately posterior to the optic chiasm, a narrow stalk called the infundibulum extends inferiorly, connecting the floor of the hypothalamus to the pituitary gland. The floor of the hypothalamus between the infundibulum and the mammillary bodies is the tubular area, and the tubular area contains nuclei that are involved with the control of the pituitary gland functions. So some of the functions of the hypothalamus contains important control and integrative centers in addition to those associated with the limbic system. The hypothalamic centers may be stimulated by sensory information from the cerebellum, the brainstem, and the spinal cord, changes in composition of the CSF and interstitial fluid, or chemical stimuli in the circulating blood that move rapidly across highly permeable capillaries into the hypothalamus, where there's no blood-brain barrier. The hypothalamus performs the following functions. So 1, the subconscious control of skeletal muscle contractions. So the hypothalamus directs somatic motor patterns associated with rage, pleasure, pain, and sexual arousal by stimulating centers in other portions of the brain. For example, hypothalamic centers control the changes in facial expression that accompany rage and the basic movements associated with sexual activity. So 2, the control of autonomic functions. The hypothalamus adjusts and coordinates the activities of autonomic centers in the pons and the medulla oblongata that regulate heart rate, blood pressure, respirations, and digestive functions. 3, the coordination of activities of nervous and endocrine systems. So the hypothalamus coordinates neural and endocrine activities by inhibiting or stimulating endocrine cells and the pituitary gland to the production of regulatory hormones. And these hormones are produced at the tubal area. They're released into local capillaries for transport to the anterior lobe of the pituitary gland. 4. The secretion of two hormones. The hypothalamus secretes antidiuretic hormone, or ADH, or vasopressin, whichever you want to call it, and oxytocin. The supraoptic nucleus produces ADH, which restricts water loss by the kidneys, and the paraventricular nucleus produces oxytocin, which stimulates smooth muscle contraction in the uterus and the mammary glands of the females and the prostate glands of males. These hormones are transported along axons that pass through the infundibulum and the posterior lobe of the pituitary gland. There are more hormones that are released into the blood for distribution throughout the body, though. And 5, the production of emotion and behavioral drives. Specific hypothalamic centers produce sensations that lead to conscious or subconscious changes in behavior. For example, stimulation of the feeding center produces a sensation of hunger. Stimulation of the thirst center produces a sensation of thirst, and the satiety center is the region that regulates food intake. These unfocused impressions originating at the hypothalamus are called drives. The conscious sensations are only a part of the hypothalamic response. For instance, the thirst center also orders the release of ADH by neurons in the supraoptic nucleus. 6, the coordination between voluntary and autonomic functions. When you think about a dangerous or stressful situation, your heart rate and respiratory rate go up and your body prepares for an emergency, and the hypothalamus makes these autonomic adjustments for you. 7, the regulation of body temperature. The preoptic area of the hypothalamus is responsible for thermoregulation, and it coordinates the activities of other CNS centers and regulates other physiological systems to maintain normal body temperature. If the body temperature of the preoptic area communicates with the vasomotor center, an autonomic center in the medulla oblongata that controls blood flow by regulating the diameter of peripheral blood vessels. In response, the vasomotor center decreases the blood supply to the skin, reducing the rate of heat loss. And finally, 8, the control of the circadian rhythm. The suprachiasmatic nucleus coordinates daily cycles of activity that are linked to the 24-hour day-night cycle. And this nucleus receives input from the retina of the eye. The output of this nucleus adjusts the activities of other hypothalamic nuclei in the pineal gland and reticular formation. So the limbic system includes nuclei and tracts along the border between the cerebellum and the diencephalon. And this system is a functional grouping rather than an anatomical one. So functions of the limbic system include establishing emotional states, linking the consciousness and intellectual functions of the cerebral cortex with the unconscious and autonomic functions of the brainstem, and facilitating memory storage and retrieval. So the sensory cortex or motor cortex and association areas of the cerebral cortex enable you to perform complex tasks, but it's largely the limbic system that makes you want to do them. For example, the limbic system is also known as the motivational system. So the amygdaloid body is commonly referred to as amygdala, and it appears to act as an inference between the limbic system, the cerebellum, and the various sensory systems. It plays a role in regulating heart rate and responding to fear and anxiety and controlling the fight or flight response by the sympathetic division of the ANS and linking emotions with specific memories. The limbic lobe of the cerebral hemisphere consists of superficial folds or gyri and underlying structures adjacent to the diencephalon. The gyri curve along the corpus callosum, which is a fiber tract that links the two cerebral hemispheres. The gyri then continue to the medial surface of the cerebrum lateral to the diencephalon. There are 3 gyri in the limbic lobe. The cingulate gyrus sits superior to the corpus callosum. The donate gyrus and the parahippocampal gyrus form the posterior and inferior portions of the limbic lobe. These gyri conceal the hippocampus to a nucleus inferior to the floor of the lateral ventricle. To early anatomists, the structure resembled a seahorse, hence the name hippocampus. And this nucleus is important in learning, especially in the storage and retrieval of new long-term memories. The fornix is a tract of white matter that connects the hippocampus with the hypothalamus. From the hippocampus, the fornix curves medially, meeting its counterpart from the opposing hemisphere. The fornix proceeds anteriorly, inferior to the corpus callosum, before curbing to the hypothalamus. Many fibers in the fornix enter the mammillary bodies of the hypothalamus. In the diencephalon, several other nuclei in the wall, thalamus, and the floor of the hypothalamus are components of the limbic system. The anterior nucleus of the thalamus relays information from the mammillary body of the hypothalamus to the cingulate gyrus on that side. The boundaries between the hypothalamic nuclei of the limbic system are often poorly defined, but experimental stimulation has outlined a number of important hypothalamic centers that are responsible for the emotions of rage, fear, pain, sexual arousal, and pleasure. And the stimulation of specific regions of the hypothalamus can also produce heightened alertness and a general excitement or generalization of lethargy and sleep. And these responses are caused by stimulation or inhibition of the reticular formation. Although the reticular formation extends the length of the brainstem, its headquarters reside in the midbrain. So the cerebrum is the largest region of the brain. Conscious thought and all intellectual function originate in the cerebral hemispheres. Much of the cerebrum is involved in processing somatic sensory and motor information, and gray matter in the cerebrum is located in the cerebral cortex and deeper in the basal nuclei. The white matter of the cerebrum lies deep to the cerebral cortex and around the basal nuclei. So a layer of cerebral cortex ranging from about 1 to about 4.5 millimeters thick covers the paired cerebral hemispheres, which dominate the superior and lateral surface of the cerebrum. The gyri increase the surface area of the cerebral hemispheres and thus the number of cortical neurons that they contain. The total surface area of the cerebral hemispheres is roughly equivalent to about 2,200 centimeters of flat surface. The entire brain has enlarged over the course of human evolution, but the cerebral hemispheres have enlarged at a much faster rate than has the rest of the brain. This enlargement reflects the number of neurons needed for complex analytical and integrative functions. And since the neurons involved are in the superficial layers of the cortex, it is there that the expansion has been most pronounced. The only solution available other than an enlargement of the entire skull was for a cortical layer to fold like a crumpled piece of paper. And so each cerebral hemisphere can be divided into lobes or regions named after the overlying bones of the skull. Your brain has a unique pattern of sulci and gyri as individuals as, say, a fingerprint, but the boundaries between the lobes are reliable landmarks. On each hemisphere, the central sulcus, which is a deep groove, divides the anterior frontal lobe from the more posterior parietal lobe, and the horizontal lateral sulcus separates the frontal lobe from the temporal lobe. This insula, or an island of cortex, lies medial to the lateral sulcus. The more posterior parietal occipital sulcus separates the parietal lobe from the occipital lobe, and each lobe contains functional regions whose boundaries are less clearly defined. Some of these regions deal with sensory information and other motor commands. I do want you to keep in mind 3 points about some of the cerebral lobes. One, each cerebral hemisphere receives sensory information from and sends motor commands to the opposite side of the body. For example, the motor areas of the left cerebral hemisphere control the muscles on the right side, and the right cerebral hemisphere controls muscles on the left side. And this crossing over has no known functional significance. The two hemispheres have different functions, even though they look almost identical. And three, the correspondence between a specific function and a specific region of the cerebral cortex is imprecise. Because the boundaries are indistinct and have considerable overlap, one region may have several functions. Some aspects of the cortical functions, such as consciousness, cannot be easily assigned by any single region. However, we know that normal individuals use all portions of the brain. What you see here is just some anatomical pictures. So the interior of the cerebrum consists mostly of white matter, and we can classify the axons of white matter as association fibers, commissural fibers, and projection fibers. Association fibers interconnect areas of cerebral cortex within a single cerebral hemisphere, and shorter association fibers are called arcuate fibers because they curve in an arc to pass from one gyrus to another. Longer association fibers are organized into discrete bundles or fasciculi, and longitudinal fasciculi connect the frontal lobe to the other lobes of the same hemisphere. Commissural fibers interconnect and permit communication between the cerebral hemispheres, and bands of commissural fibers linking the hemispheres include the corpus callosum and the anterior commissure. The corpus callosum alone contains more than 200 million axons, carrying some 4 billion impulses per second. Projection fibers link the cerebral cortex, the diencephalon, the brainstem, the cerebellum, and the spinal cord. All projection fibers pass through the diencephalon, and there, axons heading to the sensory areas of the cerebral cortex pass among the axons descending from the motor areas of the cortex. In gross dissection, the ascending fibers and the descending fibers look kind of alike. The entire collection of the projection fiber is known as the internal capsule. Moving into a little bit about the basal nuclei here. So while your cerebral cortex is consciously directing a complex movement, or solving some intellectual puzzle, other centers of your cerebrum, diencephalon, and brainstem are processing sensory information and issuing motor commands outside your conscious awareness. Many of these activities, which occur at the subconscious level, are directed by the basal nuclei. So about the anatomy of the basal nuclei here, the basal nuclei are masses of gray matter which lie within each hemisphere due to the floor of the lateral ventricle. They are embedded in the white matter of the cerebrum, and the radiating projection of fibers and the commissural fibers travel around or between these nuclei. Historically, the basal nuclei have been considered part of the larger functional group known as the basal ganglia, and this group includes the basal nuclei of the cerebrum and the associated motor nuclei of the diencephalon and the midbrain. The caudate nucleus has a massive head and a slender curving tail that follows the curve of the lateral ventricle. The head of the caudate nucleus lies anterior to the lentiform nucleus, and the lentiform nucleus consists of a medial globus, pedillus, and a lateral putnam. And the term corpus striatum has been used to refer to the caudate and the lentiform nucleus, or to the caudate nucleus and the putamen. The name refers to a striated appearance in the internal capsule as its fibers pass along these nuclei. The amygdaloid body, or part of the limbic system, lies anterior to the tail of the caudate nucleus and inferior to the lentiform nucleus. So some of the functions of the basal nuclei. The basal nuclei are involved with the subconscious control of skeletal muscle tone and the coordination of learned movement patterns. Under normal conditions, these nuclei do not initiate particular movements. But once a movement's underway, the basal nuclei provide the general pattern and rhythm, especially for movements of the trunk and proximal limb muscles. For example, when you walk, the basal nuclei controls the cycles of the arm and thigh movements that occur between the time you decide to start walking and the time you give the stop order. As you begin a voluntary movement, the basal nuclei control and adjust muscle tone, particularly in the appendicular muscles, to set your body position. When you decide to pick up a pencil, you consciously reach and grasp with your forearm, your wrist, and your hand, while the basal nuclei operate the subconscious level to position your shoulder and stabilize your arm. So how do the basal nuclei carry out their functions? So they alter the motor commands issued by the cerebral cortex through a feedback loop. Information arrives in the caudate nucleus and the putnam for sensory, motor, and integrative areas of the cerebral cortex. Lateral processing then takes place in the nuclei in the adjacent globus pedillus. Most of the output of the basal nuclei leaves the globus pedillus and synapse in the thalamus. The nuclei in the thalamus then projects the information to the appropriate areas of the cerebral cortex. Activity in the basal nuclei is inhibited by neurons in the substantia nigra of the midbrain, which release the neurotransmitter dopamine. If the substantia nigra is damaged or the neurons secrete less dopamine, basal nuclei become more active. The result is a gradual, generalized increase in muscle tone and the appearance of symptoms characteristic of Parkinson's disease. People with Parkinson's disease have difficulty starting voluntary movements because opposing muscle groups do not relax. Instead, they must be overpowered. So once a movement is underway, every aspect must be voluntarily controlled to the intense effort and concentration. So the central sulcus separates the motor and sensory areas of the cortex. And the presentral gyrus of the frontal lobe forms the anterior border of the central sulcus. The surface of this gyrus is the primary motor cortex, and neurons of the primary motor cortex direct voluntary movements by controlling somatic motor neurons in the brainstem and the spinal cord. And these cortical neurons are called paradymal cells because their cell bodies resemble little pyramids. The primary motor cortex is like the keyboard of a piano. If you strike a specific piano key, you produce a specific sound. Similarly, if you stimulate a specific motor neuron in the primary motor cortex, you generate a contraction in a specific skeletal muscle. Like the monitoring gauges of the dashboard of the car, the sensory areas of the cerebral cortex report key information. At each location, sensory information is reported in a pattern of neuron activity in the cortex. The postcentral gyrus of the parietal lobe forms the posterior border of the central sulcus, and its surface contains primary sensory cortex. Neurons in this region receive general somatic sensory information from receptors for touch, pressure, pain, vibration, or temperature. And you're aware of these sensations only when the nuclei and the thalamus relay that information to the pituitary sensory cortex. The primary sensory cortex. Sensation of light, sound, smell, and taste arrive at other portions of the cerebral cortex, and the visual cortex of the occipital lobe receives visual information, and the auditory cortex and olfactory cortex of the temporal lobe receive information about hearing and smell, respectively. The gustatory complex, which receives information from taste receptors of the tongue and pharynx, lies in the anterior portion of the insula and adjacent portions of the frontal lobe. Talking a little bit about association areas, the sensory and motor regions of the cortex are connected to nearby association area regions of the cortex that interpret incoming data or coordinate motor responses. Like the information provided in the gauges in a car, arriving information must be noticed and interpreted before the driver can take the appropriate action. So sensory association areas are cortical regions that monitor and interpret information that arrive at the sensory areas of the cortex. Examples include the somatic sensory association area, the visual association area, and the auditory association area. The somatic sensory association area monitors activity in the primary sensory cortex, and the somatic sensory association area allows you to recognize touch as a light mosquito landing on your arm and gives you a chance to swat that mosquito before it bites you. The special senses of smell, sight, hearing involve separate areas of sensory cortex, and each has its own association area. These areas monitor and interpret arriving sensations. For example, the visual association area monitors patterns of activity in the visual cortex and interprets those results. So you'll see the symbols C, A, and R when the stimulation receptors in your eye, which then leads to the stimulation in your neurons in the visual cortex. And once your visual association area recognizes those letters, you'll get the C plus A plus R will then equal the word car. So an individual with a damaged visual association area could scan the lines of a printed page and see the rows of symbols that are clear, but wouldn't perceive any meaning from the symbols. Similarly, the auditory association area monitors sensory activity in the auditory cortex. Word recognition takes place in this association area. So the premotor cortex or somatic motor association area coordinates learned movements, and the primary motor cortex does nothing on its own, any more than a piano keyboard can play itself. So the neurons in the primary motor cortex must be stimulated by neurons in other parts of the cerebrum. And when you perform voluntary movements, the premotor cortex relays the instructions to the primary motor cortex. And with repetition, the proper pattern of stimulation becomes stored in your premotor cortex. And you can then perform the movement smoothly and easily, triggering the pattern rather than by controlling the individual neurons. And this principle applies to any learned movement, from something as simple as picking up a glass to something as complex as playing the piano. One area of the premotor cortex and the frontal eye field controls learned eye movements, such as when you scan the lines of, say, words on a computer. Individual with damage to the frontal eye field can understand written letters and words, but can't read because their eyes can't follow the lines on a printed page. Integrative centers are areas that receive information from many association areas and direct extremely complex motor activities. And these centers also perform complicated analytical features. For example, consider the prefrontal cortex of the frontal lobe. It integrates information from the sensory association areas and performs abstract intellectual functions, such as predicting the consequences of possible responses. Integrative centers are located in the lobes and the cortical areas of both cerebral hemispheres. And integrative centers concerned with complex processes such as speech, writing, mathematical computation, and understanding spatial relationships are largely restricted to either the left or the right hemispheres. And these centers include the general interpretive area and the speech centers. The corresponding regions on the opposite hemisphere are also active, but their functions are less well defined. The general interpretive area, also called the Wernicke's area, is present in only one hemisphere, typically the left. And this analytical center receives information from all sensory association areas and plays an essential role in your personality by integrating sensory information and coordinating access to complex visual and auditory memories. Damage to the general interpretive area affects your ability to interpret what you see or hear, even though you understand the words as individual entities. For example, if your general interpretive area were damaged, you might still understand the meaning of the spoken words and the spoken word sit and hear, because word recognition takes place in the auditory association areas. But you will be totally confused by the request to sit here. Damage to other portions of the general interpret area might leave you able to see a chair clearly and to know that you recognize it, but you would be unable to name it because the connection between your visual association area has been disrupted. So in the speech center, some of the neurons in the general interpretive area innervate the speech center, also called the Broca's area or the motor speech area. This center lies along the edge of the premotor cortex in the same hemisphere as the general interpretive area, which is again usually the left. The speech center regulates the pattern of breathing and vocalization needed for normal speech, and this regulation involves coordinating the activities of the respiratory muscles and the laryngeo and pharyngeo muscles, and the muscles of the tongue, the cheek, the lips, and the jaw. A person with a damaged speech center can make sounds but not words. So the motor commands issued by the speech center are adjusted by feedback from the auditory association area, also called the receptive speech area. Damage to related sensory areas can cause a variety of speech-related problems, and some infected individuals have difficulty speaking, although they know exactly which words to use. Others talk constantly, but use all of the wrong words. The prefrontal cortex of the frontal lobe coordinates information relayed from the association areas of the entire cortex. In doing so, it performs such abstract intellectual functions as predicting the consequences of events or actions. The prefrontal cortex doesn't fully develop until the early 20s, and damage to the prefrontal cortex leads to difficulties in estimating temporal relationships between events. Questions such as how long ago did this happen or what happened first become difficult to answer at that point. The prefrontal cortex has extensive connections with other cortical areas and with other portions of the brain. Feelings of frustration, tension, and anxiety are generated as the prefrontal cortex interprets ongoing events and makes predictions about future situations or consequences. If the connections between the prefrontal cortex and other brain regions are physically severed, the frustrations, the tensions, and anxieties are removed. During the middle 20th century, this rather drastic procedure called a prefrontal lobotomy was used to, quote unquote, cure a variety of mental illnesses, especially those associated with violent or antisocial behaviors. After a lobotomy, the patient would no longer be concerned with what had previously been a major problem, whether psychological, being hallucinations, or physical, being severe pain. However, the individual was often equally unconcerned about the tact, decorum, and toilet training. And since then, drugs that target specific pathways and regions of the CNS have been developed, so lobotomies are no longer used to change behaviors. Speaking about Brodmann's areas, in the early 20th century, numerous researchers attempted to describe and classify regional differences in the histological organizations of the cerebral cortex. They hoped to correlate the patterns of the cellular organizations with those specific functions. But by 1919, at least 200 patterns had been described, but most of the classification schemes had since been abandoned. However, the cortical map prepared by Corbani and Brodmann in 1909 has proved useful to neuroanatomists. Brodmann, who was a German neurologist, described about 50 patterns of cellular organization in the cerebral cortex. And some corresponded to known functional areas. For example, the Brodmann area 44 corresponds to the speech center, and area 41 to the auditory complex. Area 4 follows contours of the primary motor cortex. In other cases, the correspondence is less precise. For instance, the Brodmann area 42 forms only a small portion of the auditory association. Again, just a table summarizing what we just talked about. Speaking on hemispheric lateralization, each of the two cerebral hemispheres is responsible for specific functions that are not ordinarily performed by the opposite hemisphere. This regional specialization is called hemispheric lateralization. In most people, the left hemisphere contains the general interpretive and speech centers and is responsible for language-based skills. For example, reading, writing, speaking, depend on processing done in the left cerebral hemisphere. In addition, the premotor cortex involved with the control of the hand movements is larger on the left side for the right-handed individuals than for left-handed ones. The left hemisphere is also important in performing analytical tasks such as mathematical calculations and logical decision-making. And for these reasons, the left cerebral hemisphere has been called the dominant hemisphere. The right cerebral hemisphere analyzes sensory information and relates the body to the sensory environment. Interpretive centers in this hemisphere permit you to identify familiar objects, by touch, smell, sight, taste, or feel. And for example, the right hemisphere plays a dominant role in recognizing faces and understanding three-dimensional relationships. It is also important in analyzing the emotional context of a conversation. For instance, distinguishing between the threat, get lost, or the question, get lost. Individuals with damaged right hemisphere may be unable to add emotional inflections to their words. So, left handed people represent about 9% of the human population, and in most cases, the primary motor cortex of the right hemisphere controls motor functions for the dominant left hand. However, the centers involved with speech and analytical function are in the left hemisphere, just as they are in right handed people. Interestingly, an unusual high percentage of musicians and artists are left handed. The complex motor activities performed by these individuals are directed by the primary motor cortex and association areas of the right cerebral hemisphere, near the association areas involved with spatial visualization and emotions. Various methods have led to maps of the brain activity and brain functions, and the primary sensory cortex and the primary motor cortex have been mapped by direct stimulation in patients undergoing brain surgery. The functions of other regions of the cerebrum can be revealed by behavioral changes that follow localized injuries or strokes. The activity of specific regions can be visualized by a PET scan or through functional magnetic resonance imaging or an fMRI. Clinicians commonly monitor the electrical activity of the brain to assess brain activity. Neurofunction depends on electrical events of the plasma membrane of neurons. So the brain contains billions of neurons, and their activity generates an electrical field that can be measured by electrodes that are placed on the brain or the outer surface of the skull. And the electrical activity changes constantly as nuclei and cortical areas are stimulated or quiet down. A printed recording of the electrical activity of the brain is called electroencephalogram, or an EEG, and the electrical patterns observed are called brain waves. Alpha waves occur in the brain of healthy, awake adults who are resting with their eyes closed. Alpha waves disappear during sleep, but they also vanish when the individual is able to concentrate on specific tasks. During attention to stimuli or tasks, higher frequency beta waves replace alpha waves, and beta waves are typical of individuals who are either concentrating on task, under stress, or in a state of psychological tension. Theta waves may appear transiently on sleep in normal adults, but are most often observed in children and intensely frustrated adults. The presence of theta waves under other circumstances may indicate the presence of brain disorders such as a tumor. Delta waves are large amplitude waves, low frequency waves, and they are normally seen during deep sleep in individuals of all ages. Delta waves are also seen in the brains of infants whose brains are still developing, and awake adults when a tumor, a vascular block, or inflammation has damaged portions of the brain. Electrical activity in the two hemispheres is generally synchronized by a pacemaker mechanism that appears to involve the thalamus. An asynchrony between the hemispheres can indicate localized damage or other cerebral abnormalities. For example, a tumor or injury affecting one of the hemispheres typically changes the pattern in that hemisphere, and the patterns of two hemispheres are no longer aligned. A seizure is a temporary cerebral disorder accompanied by abnormal movements, unusual sensations, inappropriate behaviors, or some combination of these symptoms. Clinical conditions characterized by seizures are known as seizure disorders or epilepsies. Seizures of all kinds are accompanied by a marked change in the pattern of electrical activity recorded in electroencephalogram. The change begins in one portion of the cerebral cortex, but may then spread across the entire cortical surface like a wave across the surface of a pond. So the nature of the signs and symptoms produced depends on the region of the cortex involved. If a seizure affects the primary motor cortex movements will occur. If it affects the auditory cortex, the individual will hear a strange sound.